Now it is my pleasure to introduce today's first speaker, Dr. Elias Friman. Elias did his PhD at the University of Lausanne in the lab of David Suter, where he did some really nice work on the role of OCT4 and SOX2 in regulating chromatin accessibility and cell fate across the cell cycle in embryonic stem cells. For his postdoc, he then moved to Edinburgh to join the lab of a past Fragile Nucleosome speaker, Wendy Bickmore. And today he's going to be presenting his postdoc work on ultra long-range interactions between active regulatory elements. So please join me in giving a warm, albeit virtual, welcome to our first speaker, Elias Friman.
Thank you very much, Ben. Let me do this again. Is this working now?
Yes, that looks perfect.
Okay, great. Thank you so much for the introduction and for giving me the chance to present our work here. So today we're going to talk about ultra long range interactions in chromatin. So I'll just do a brief introduction. I'm interested in the organization of chromatin and particularly the mechanisms that regulate how chromatin is organized and how these relate to particularly transcription. And we know already many things about how this works. I'm just listing some of the things here. This is not an exhaustive list. So one of the most studied mechanisms is cohesin-mediated loop extrusion, where the cohesin loads onto chromatin and kind of creates this looping out. And then it reaches some barrier elements, usually CTCF sites. And this leads to the formation of TADs, as you may have me be aware of, but also kind of focal interactions between these CTCF sites. And the scale of these interactions are up to about 1 megabase, because it will encounter these barriers otherwise if it goes further out. And on the larger scale, we also have polycomb interactions. So regions in the genome that are bound by polycomb can both interact in local domains, but also with each other across very large distances, several tens of megabases apart. And even larger than that, we have these compartments, which are much bigger genomic regions. There are several hundred kb, several megabases in some cases, and they interact with their own compartments. So if you're in the A compartment, which is essentially euchromatin, then you tend to interact with your A compartment, other A compartments and so on. And this occurs both within and between chromosomes. So the point here is just to say there are different mechanisms and there are different scales that they operate at. So before we move on, I just want to be a quick description of the kind of data I'm looking at. So we're using this technique called Hi-C or micro-C, which is very similar. So the idea here is that you cross-link your chromatin and then you do a proximity ligation and then you sequence a junction. So it means that you will sequence things that are sequenced together are near each other in space. That's the idea. And the proportionality between how much you sequence them and how often they are nearby in space. And after you process your data, you get something like this, where the diagonal represents regions. And then if you pick two regions and look at the intersection between them, you will then kind of read out how much they interact. And that will be then the color of this map. And then you can see different structures here, the kind of things I've been talking about. And one problem with this kind of data is you might see here, for example, that there's a lot of sparsity. There's empty regions. So one way to get away from this problem, which comes from the fact that the genome is very large and you need to sequence a lot to really cover everything, is doing averaging. So we use this technique called pileups, which is essentially you pick a set of regions. So here in the schematic, we have 5 regions, and you take all the different combinations of those 5, so all the pairs, and then you look at the interaction between them, and then you average them up to get an average interaction between this set. So for example, for polycomb sites, you will, in the graph that I will show a lot of these, you will basically have one set of polycomb sites on the left and one set of polycomb sites on the bottom, and they interact here in the middle. And this is the average enrichment for this set of regions. So what we asked in this project was what factors are associated with interactions between cis regulatory elements at different scales. So cis regulatory elements means, transcriptional regulatory elements such as enhancers and promoters. And scale here, I'm referring to the distance between the regions. So essentially, if two regions are close by on the genome and they tend to interact, what tends to be bound there? And if they are far apart and they tend to interact, what binds there? So that's what we wanted to see. So the approach here, I'm just going to hide this thing. The approach here was basically taking a set of cis-regulatory elements in mouse embryonic stem cells, and we split these pairs because you have all the different pairs between them. Those that are short range, which here is 0.1 to 1 megabase apart, and the long range, which is 1 to 10 megabases apart. And then you take all these pairs, and then you look in these, or we looked in these two databases, which have a lot of ChIP-seq binding profiles, and we basically split them into overlapping and non-overlapping. And then you compare the contact frequency between these sets. So for example, ring 1b, which is a polycomb component, remember polycomb can form these contacts. If you have ring 1b bound at both sides, then you have an increased contact frequency compared to if you don't have polycomb bound on both sides. And then we did this kind of analysis for all the different data sets, and we get some p-value and an effect size. So I'm going to show you one plot from this analysis, and it's this one. So you have the y-axis, which represents the enrichment on the short range, and the x-axis, which represents the enrichment on the long range, and higher than 0.5 means enriched. And you may see here at first glance, you have this cluster of yellow at the bottom. So these are enriched for short range, but not long range. And these are the cohesin factors. And then you have the blue dots, which are the polycomb components. They are enriched very highly, and also both at short and long range. So now if we just visualize this again using the pileup method, we see here that if we split the pileups by distance, you see that the RAD21, which is a cohesin component, you see interaction between these at short range, but not long range, and you see interaction between ring 1b sites across these ranges. And then we saw all these red and green dots, which we've kind of roughly classified as transcription factors and transcription cofactors, so things that are associated with active transcription. We saw these to be quite enriched for long-range interactions. So if we then look at these in the same way, we see, for example, CFP1, which is a CpG island binding protein, KLF4, which is a transcription factor. We see these interactions at really large distances, 1 megabase up to 100 megabases apart. And we call these ultra long range interactions. And we were quite interested to understand what's going on here. And I just want to make a point about the enrichment. So the enrichment here is versus what you expect, which already takes into account the distance. So it's not that this bin here of the very far apart ones interact as much as the ones that are closer together, but they are similarly enriched to what you would expect, which you can also see from the neighboring regions here. So what's going on here? So we looked at this CpG islands because we had enrichment for CFP1, which binds to CpG islands. And if you split these CpG islands by their density of CpG, you see a correlation between the CpG density and the ultra long range interactions and interactions in general. And similarly, at transcription start sites, you see a correlation between expression and ultra long range interactions, and these non-expressed genes, they do not tend to interact. Okay, so we just wanted to make sure that it's really nothing to do with polycomb already or cohesin. So we looked in data sets where, in this case, RAD21 has been degraded, and you lose your CTCF interactions, but you do not lose your ultra long range interactions that we see here. And similarly, between the ring 1b binding sites, you lose the polycomb interactions if you knock out ring 1b, but we don't lose these ultra long range interactions. So this is a third set of interactions between cis regulatory elements that is different from polycomb and cohesin. And because we see these very large distances, we also wanted to see, couldn't we even see enrichment between chromosomes between these elements? And indeed we can. I mean, again, this is very sparse data, so I'm not saying that this is extremely rare, but nevertheless enriched, meaning that they are more likely to interact with each other across chromosomes than the other regions on those chromosomes. So we also looked at enhancers. Do they interact in the same way? So these are H3K27 acetylation peaks, which do not overlap with polycomb sites. They do not overlap with CpG islands, and they don't overlap with transcription sites. So these are putative enhancers. And we see, again, a scaling with activity. And here we're using the H3K27 acetyl signal as a proxy for activity. So this also occurs between enhancers. And I'm not showing it here, but it's also the case that enhancers interact with promoters, promoters, enhancers, and enhancer-promoter interactions occur across these very large distances. So can these things be dynamic? And also, do we just see them in mouse embryonic stem cells? Is this some weird phenomenon that's specific to these? This is not specific to mouse embryonic stem cells because, for example, here, we use these data recently published in the preprint, where they take macrophages and they stimulate them, and they do a time series of K27 acetylation and high C. And we can see here that within a few hours in these regions, for example, you have a few hundred which robustly gain acetylation. And you can see here within the few hours, you also gain interactions. This is rather noisy because there's not that many regions to work with. And similarly here, even though these don't interact much to begin with, they also tend to decrease in interaction in line with their decrease in K27 acetylation. So it's something that occurs in other cell types, and it can be quite dynamic. We then looked in zebrafish, and we picked out regions that are specific to brain in terms of their H2K27 acetylation level. And we can see here that the brain enhancers interact in the brain, but not in the muscle, as we would expect from this specificity. So obviously you have more 27 acetylation because that's what we picked on. But we also found this increase in DNA methylation. So we thought perhaps the interaction is somehow mediated by the fact that you have, obviously in these vertebrate genomes, you have methylation everywhere except where you have demethylation, for example, at CpG islands or also at these enhancers. And maybe if you gain methylation, you will then lose the interaction. So to see if this has anything to do with methylation, we had a look in the Drosophila genome because Drosophila doesn't have any kind of DNA methylation. And surprisingly, we found the similar type of long-range interactions in Drosophila. Here again, scaling with expression, and the scale here is smaller because they have a shorter chromosomes. But nevertheless, we see these very long-range interactions also between K27 acetylation peaks that don't have polycomb. So this does not seem to be a vertebrate-specific phenomenon and not related to DNA methylation. So if it's not related to that, perhaps it's related to transcription or somehow something related to transcription. If we take data where cells have been treated with triptolide or flavopyridol, which blocks transcription at different steps, so these are small molecules, at least within these 45 minutes, you don't see a loss of interaction. So at least it's not something that's immediately downstream of transcription. And you can even see here when you degrade RPB1 using a Degron system, so this will basically, you will lose the whole Pol2 complex. And you see that you still have these interactions, even without Pol2. So I haven't shown you, but in mitosis, as you may expect, we have a loss of interactions, and then they have to reform directly after mitosis. So maybe if you come out of mitosis without RNA polymerase II, they can't be reformed. But again, surprisingly, you don't need RNA polymerase II after mitosis to reform these contacts. So here's the untreated and here's the RPB1 degraded after mitosis. Okay, we also looked at BRD4 degrons. Doesn't seem to affect these ultra long range interactions. And also there's different mediator perturbations, which also do not seem to affect these interactions. So finally, we thought, okay, maybe it has something to do with this acetylation then. So P300 binds to these regions and puts the acetyl mark everywhere. And if you block the activity of P300, which they have done in this paper, you see that you have this loss of H2K27 acetylation at the peaks, but also globally on chromatin. So maybe at this point, you will lose the interactions. But again, surprisingly, these interactions, at least after this 3 hour of perturbation, we don't see any loss of interactions, even if you don't have P300 activity and you don't have H2K27 acetylation or much lower levels. So finally, we can say that we see these ultra long range interactions between active regulatory elements. They occur in metazoan species, both vertebrates and invertebrates. They're dynamic between tissues and also even on shorter timescales. They're associated with CpG islands, even though you don't need a CpG island to have them. They're associated with transcriptional activity and K27 acetylation. but they don't appear to be dependent on polycomb cohesin, DNA methylation, transcription, BRD4 mediated P300, or H3K27 acetylation. So just to end up here, what we propose is basically that you have three modes of interactions between cis regulatory elements. You have the loop extrusion, you have polycomb, and then you have this tendency of active regions to interact with each other through some mechanisms which we have not uncovered. And this occurs over very, very large distances. And if we just look at the kind of plot here, what we see is an interaction in the center, which represents the fact that the two, the red triangles here in this case, they tend to interact with each other. But also the stripe you see here between means that the individual regulatory elements also tend to interact with the regions around, which is basically the other A compartment, which is very far away. So these are the two things. And I haven't made any functional claims here, and I'm not going to, but you could speculate perhaps that this increase in interaction frequency, not between any two given elements, but between the cumulative increase in interactions between all the elements when they are highly transcribed, could contribute to their transcriptional activity, assuming that these are functional contacts. So with that, I would like to finish and thank my lab, and particularly Wendy, for the support. Also, big thanks to Ilya, who has been working with me on this. And then also to Sheila and Hannes, and also the Swiss National Science Foundation for funding my postdoc. And thank you very much for your attention.
Thank you, Elias. That was fantastic. So just a quick reminder, you can type your questions into the Q&A box or raise your hand and we'll unmute you to ask a question. And so I'll start with some of the questions in the Q&A box. So Felix Jonas says, great talk. I was wondering if you can induce colocalization by expression or acetylation so that it's not required, but maybe sufficient?
Well, it's hard to test. I mean, I guess the closest thing was this macrophage stimulation, because the problem is, because of the sparsity of the data and the low frequency of interaction between any two regions, I wouldn't expect that, for example, if you recruit, I actually looked at some data that have done this, like if you recruit something to one specific place, you don't necessarily see an increase, but it's probably just a problem of resolution and so on. I mean, I would expect so, indeed. I don't think it's because when you do, if you pull down P300 to a region, for example, you have a lot of things happening. So it wouldn't only be acetylation, you would also recruit a lot of other stuff. So it's hard to kind of disentangle. But I would think that you would need to do it in many places to see that, it.
Yeah, it'll be interesting. Something we talk a lot about is like what drives establishment versus maintenance of these types of things.
I wonder if I wonder if there's a distinction here, because I imagine, at least in this case, that because they are so dynamic, probably because these are things that kind of just bump into each other, I doubt that there's really an establishment where I think they are just it's just something that happens. If so, there's just a constant maintenance. And then of course in mitosis you have things changing, but then it just kind of keeps going again. I don't think these are stable contacts, so to speak.
Yeah. Have you tried any sort of imaging to get a sense of the frequency with which these are interacting?
Yeah, we are doing that now. But it's a bit hard because, well, it's not hard. You can, of course, pick regions that you can see in your high C, but again, as most of these, if you pick things very far apart, you wouldn't expect them, any given two regions, to interact very often. It's more the accumulation. But indeed, we are testing some of these things that we know are not driven by polycomb and cohesin, and to see that they are interacting more often. And I expect the absolute frequency to be quite low, but higher than the regions next to them. That's the most important.
Another question is, when you looked at short range versus long range interacting factors, have you considered classifying transcription factor and cofactors depending on which interact with SMCs versus those that interact with polygon group proteins? If you classify recruiting factors through the mechanism.
I haven't done that. So I guess what you would see is if something is kind of bound to the same regions that polycomb is, then yeah, you would see that they kind of go with the polycomb. I haven't done that. So that might be a good idea. But in general, with those red and green dots that I showed on the first plot, they correlate very strongly with having with the number of transcription sites they overlap with. So I think most of them are more associated with activity, but it's true that some of them will be related to cohesin and to polycomb as well.
So Charles has his hand up.
Yeah, very nice talk. I think your suggestion that these actually reflect compartments makes a lot of sense to me. And there's at least some literature showing that compartments can be quite stable across different perturbations. And so I guess the question is, these perturbations, how long do they run? And is there value in maybe collecting a data set where you do one of these perturbations across a much longer time? To see if there is some new steady state that things sort of come to.
Yeah, so yeah, I thought a lot about this. So I'm not saying that they are compartments. I think they are. I mean, it's a bit of semantics in some point, because compartments, we kind of don't really know how they're formed, and indeed, as you say, we can't really perturb them. So in that sense, this is very similar to compartments. And there have been some other preprints out that kind of, I think they see something similar. And but I think the problem I see with the suggestions that if you do these longer term perturbations, I mean, I would expect things to change at some point, but then it's anyway won't tell you what it is that's changing, because if you wait long enough, then something a lot of things will happen. I guess that's the point, is that, for example, if you remove acetylation for many more hours, then things will just start to shut down and you have indirect effects. It won't really tell you what's driving it. So I think that's the problem with that.
Yeah, I think that's a great answer. And I mean, to some extent, that is your cross cell type comparison that you've already made. Yeah, I think that's, well, great talk.
Thank you.
All right. I think in the interest of time, we'll wrap it up there. Elias, there are many more questions in the Q&A box. So if you have, if you want, you can type in your answers there for the ones that we were not able to ask live.
Great. Thanks a lot.
Really a very thought-provoking talk and generate much interest. So thank you again. I'll pass it on over to Matteo to introduce our second talk.